Hello and welcome to the lecture on placentation and fetal circulation. This lecture focuses on the formation of the placenta and the uteroplacental circulation. Then we will look at some of the shunts that are visible within the fetal circulation. After this lecture and any further necessary study, you should be able to give an account of the functions of the placenta, give an account of the formation of the maternal and fetal components of the placenta, and describe the appearance of the placenta at birth. Describe the placental circulation with particular reference to the separation of the maternal and fetal circulations. And give an account of the normal fetal circulation and give examples of problems arising when fetal shunts fail to close after birth. Before we get underway, let's have a look at this placenta. Here we have a newborn with the white umbilical cord. Now that the blood is no longer being pumped within that cord, it has a white appearance. To the right of this image we could see the placenta. More specifically, we could see the embryonic side of the placenta. The placenta has two surfaces, the maternal side and the embryonic side. Each side has a different feature that we will come on to discuss later. But note how centrally that this umbilical cord inserts onto the embryonic side of the placenta. This is really important. As a clinician, your role after the delivery of the baby and the placenta will be to inspect the placenta and ensure it is intact. Retaining any placental parts could have devastating consequences, either directly to the pregnant person or to any subsequent future pregnancies. So taking us back to basics, what is the placenta for? Well, this is where we are going to exchange nutrients and gases. The fetus is wrapped deeply within the uterus and doesn't have access to air for breathing nor food for eating. So getting oxygen and nutrition to the fetus must happen in order for that fetus to survive and develop. Additionally, the placenta is required for hormone production. Once that corpus luteum has fulfilled its role in maintaining the hormone production throughout early pregnancy, the placenta must take on the role of driving hormone secretion. Major changes within the developing placenta take place around week 9 that allow for exchange across a very thin membrane. It increases its surface area, which increases the efficiency of exchange, and it achieves this by the growth of villi. Components of the placenta, if you remember, are derived from the embryonic tissues and are not derived from maternal tissues. So the placenta is driven by the genes that were once present in the zygote immediately after fertilization. When we look right back to the developing embryo, around week 2 at the embryonic stage, we saw how the morula developed into a blastocytic cavity and transformed into a blastocyst. The inner cell mass of the blastocyst went on to form the embryo, whilst the outer cell mass went on to differentiate into trophoblasts. Those trophoblasts and the extra embryonic mesosome will go on to form the supportive structures, the placenta and the umbilical cord. Those trophoblasts differentiate further and invade the maternal endometrium of the uterus and erode the vessels within the endometrium and establish a uteroplacental circulation. The vessels that they are establishing a blood supply with are from the spiral arteries of the uterus. Now it goes without saying, but the development of the placenta is vital. As the fetus does not have external access for gas and nutrient exchange, the placenta plays a key role in providing these substances for the developing fetus. If the placenta develops poorly, the risks to the fetus are high. If the placenta develops incorrectly, not only is the fetus at risk, but also the pregnant person is at risk too. This is something we will look at more closely in the practicals when we consider the conditions such as preeclampsia and placenta accreta. So we've talked about gas exchange happening at the placenta, but what does that actually look like? Well, not only is it vital to get the good things into the fetus, but it's also essential to get the waste products or the metabolites out of the fetus too. So let's have a look at this list. On the left we can see oxygen is reaching the fetus. Whilst on the right we can see carbon dioxide is being sent back to the placenta, where it will re-enter maternal circulation and be expelled. We have the placenta releasing metabolites from the fetus such as urea, uric acid, creatinine and bilirubin, releasing these back into the maternal circulation for the body to eradicate them in the conventional manner. We have electrolytes, nutrients, vitamins and trace elements being delivered to the fetus, and hormones and antibodies. But what we also have, and some which can be problematic, are things such as drugs. These could be therapeutic or also recreational. Some of these we know to be beneficial, for instance therapeutic steroids for lung maturity, but we know that some can be harmful, like recreational drugs or teratogenic prescription medications such as isoretinoids or SSRIs. We know that alcohol and some viruses are also capable of crossing the placenta and being delivered to the fetus. So although the placenta is vital for life, some of the properties of the placenta mean that not only the good stuff is passed to the fetus, some harmful or teratogenic substances are also transmitted this way too. So my favourite cells are these syncytia trophoblasts, not because their name is lovely to say, but also because these cells are very busy little bees. The cells are hugely important for helping with implantation and burrowing into the endometrium, but they also play a role in immune suppression, and as we'll discuss in a short while, establishing a utero-placental circulation. In addition to these roles, these little worker bees are fundamental to hormone production and secretion to support and hide the pregnancy. One of these hormones we exploit for testing pregnancy, and that is HCG, or human chorionic gonadotropin. HCG is secreted to support the corpus luteum and modulate the maternal immune response. The corpus luteum's role is to maintain pregnancy until the placenta can take over. At the 4th month of pregnancy, the placenta takes over from the corpus luteum and begins to produce progesterone. Estrogen is important for uterine growth and breast development, and assisting with the latter is a hormone called somatomamotropin. Both of these hormones are also produced by the syncytia trophoblasts within the placenta. So let's recap the cellular journey we have been on so far. Our fertilised oocyte is now called a zygote. That undergoes rapid mitotic division called cleavage until it reaches a 16 to 32 cell ball, for which we now call it a morula. After the morula is formed, the ball of cells develops a blastocytic cavity, and we now call the whole unit a blastocyst. The cyst forces the inner cell mass to one side. These are construed of embryoblasts. The ones we are interested in today, though, are the ones that are on the left. These are the outer cell mass, which are construed of trophoblasts. These trophoblasts go on to differentiate into cyto and syncytio trophoblasts. But the question remains, how do we go from this to this? How does this ball of cells with a developing embryo end up being a fully functioning placental organ? So at day 7 we can see the beginnings of implantation. We can see our bilaminar disc made up of epiblast and hyperblast cells. We can see how this dorsally embeds onto the surface of the endometrium. Those trophoblasts are differentiating into darker green cytotrophoblasts and the paler multinucleated syncytiotrophoblasts. It is worth pointing out that the syncytiotrophoblasts have got their name from the syncytium that they have produced. They have lost their cellular membranes between each nuclei, so are now pooling that nuclei in a syncytium. The lack of cell membranes forms an important role in hiding the embryo from the maternal immune system. Even if the maternal immune system was to detect antigens from the embryo, the immune cells would have difficulty reaching the embryo because of the lack of cellular membranes from the syncytiotrophoblasts. If we have a look at day 9, we can see that we are fully implanted. The blastocyst has made its way entirely into the endometrium and has formed a little scab on the cavity surface. This is known as a fibrin or coagulation plug. We can see the bilaminar disc is now beginning to form a primary yolk sac from that migrating hyperblast layer. We can also see the epiblast layer has formed a cavity of its own, the amniotic cavity. This will become very important when we look at twinning in future lectures. We can see the syncytial trophoblasts surrounding the embryo. These have been the ones responsible for the burrowing and the anchoring of the embryo in the uterine lining. Additionally, these cells are working on eroding the maternal capillaries and forming the beginnings of a uteroplacental circulation. They form these lacunae, which are little lakes of maternal blood flow. At this point, we don't have a circulation in the embryo, so instead, simple diffusion allows nutrients and oxygen to make its way from these lacunae to the developing cells. We are also able to get substances from the enlarged uterine gland by simple diffusion. Once full implantation occurs, we have something called a decidual reaction, which modifies the uterine lining to allow for exchange. The decidual reaction packs the uterine lining with nutrients, and it encourages the engorgement of the maternal capillaries. In some texts, you may see this being referred to as maternal sinusoids. If we look at days 10 and 11, we can see some more structures have begun to develop. Our primary yolk sac has formed from the hyperblast layer, and the amniotic cavity in the epiblast layer has also enlarged. The trophoblastic lacuna has started to enlarge and bleed into each other. Within the layer of the cytotrophoblasts in the neon green, we can see the formation of extra embryonic mesoderm, which has begun to envelope the entire embryo. We can see the syncytial trophoblasts have degraded the wall of the maternal capillaries, allowing the blood to pool into those lacunae. On this slide, we are moving from where we left off on day 10 to 11 and are moving towards the middle picture at days 11 to 12. We can see the extra embryonic mesoderm derived from the cytotrophoblasts and the hyperblasts has made its way around the entire embryo. As we continue to day 12 to 13, that extra embryonic mesoderm is developing another fluid-filled cavity called the chorionic cavity. Moving forward in development to the first image, we can see our bilaminar disc. Remember, gastrulation doesn't occur until the 3rd week. But this time we can see that the primary yolk sac is currently being displaced by a second round of hyperblast migration and the formation of the definitive or the secondary yolk sac. Looking at the extraembryonic mesoderm, we can see the chorionic cavity has increased in size and is pinching at the extraembryonic mesoderm dorsally. If we look at the syncytial trophoblasts, we can see that the lacunae are beginning to coalesce together, and by day 14 to 15, we end up with the image on the bottom right. The chorionic cavity has expanded, and now the only things connecting the embryo to the complex is a thin strip of extraembryonic mesoderm called the connecting stalk. This stalk will go on and form our umbilical cord. And that brings us to the end of what we have talked about earlier in medicine, which will provide you with a nice background for what we are about to build on today. If we look at where we left off on the last slide, let's have a closer look at the cyto and the syncytial trophoblasts. The cytotrophoblasts are the ones in darker green, and they have a nice boundary formed by cellular membranes, whilst the syncytial trophoblasts are the rugged frills around the outside and are multinucleated through the loss of their cell membranes. Once we get to this stage in development, we can zoom in on these cells, which is what the image on the right demonstrates. We can see the formation of something called primary villi. The extraembryonic mesoderm signals to the cytotrophoblasts and encourages them to proliferate locally and form projections into the syncytial trophoblasts. So what essentially happens is the primary villi is formed by syncytial trophoblast core with a covering of with a cytotrophoblast core and a covering of syncytial trophoblasts. These primary villi are going to extend into the lacuna, and some texts will refer to these lacuna now as being intervillous spaces. That primary villi needs to develop further because it's still not very good at getting nutrients to the developing embryo, who is in more demand of nutrition. What happens now is more modifications are made to increase the surface area and develop those primary villi into secondary and then finally tertiary villi. So what happens to the primary villi to help them mature into secondary villi? Well, we have the invasion of extraembryonic mesoderm. So the secondary villi are constructed of an extraembryonic mesoderm core with a thin layer of cytotrophoblasts and a thin cap of syncytial trophoblasts penetrating into the lacuna or the intervillous spaces. As these secondary villi develop further, the extraembryonic mesoderm gives rise to blood vessels, which will connect to the embryo proper. These blood vessels will allow for exchange of gases and nutrients and metabolites, all of the things we looked at towards the beginning of the lecture. Once these vessels develop, the tertiary villi are formed, and we have a working uteroplacental circulation. What is important to understand is that there is no mixing of fetal and maternal blood. The lacuna are filled with maternal blood, and the extraembryonic vessels are full of embryonic blood. These vessels and the lacuna come in close proximity, which allows for exchange to happen across the membranes, but blood does not cross these membranes, which is a really significant concept to grasp, as mixing of blood could be devastating. And that's how we moved from that newly implanted embryo into a fully formed uteroplacental circulation. In this image, we could see the tertiary villi implanted into the endometrial lining. We can see the maternal spiral arteries leaking into the intervillous spaces and providing a pool of blood close to the vessels of the placenta and allow for nutrient and metabolite exchange. We can see that the extraembryonic vessels are travelling within an umbilical cord and branching out to form the placenta to transport those nutrients from the tertiary villi all the way back to the developing fetus. Later on in pregnancy, the tertiary villi will go on to form free villi, which increases the surface area even more, as the requirements for the fetus increase. So this is an example of what I would advise you to do. Here are the step-by-step images that I have drawn out, and this is a really good way of picturing the modifications and the developments we have seen. Pause the video if you are wanting to take inspiration from these pictures to create your own. And likewise on these slides. Pause the video or screenshot for sketching out your own revision sources. What we can see on here is how we transition from the secondary villi into the tertiary villi, with its chorionic vessels. We haven't discussed this up to this point, but what we can see on this drawing on the right is a plate called the decidual plate of the uterine lining. In normal circumstances, this is the depth at which the syncytial trophoblast should penetrate to. In the event that these cells permeate deeper than the decidual plate, we develop something called placenta accreta. This is problematic because when it comes to delivering the placenta after birth, the pregnant person can undergo extreme hemorrhage. Sometimes, in extreme circumstances, the syncytial trophoblast can penetrate much deeper and through the entire uterine wall and invade some of the pelvic abdominal organs. In the event that this plate isn't reached, preeclampsia may develop. The placenta is poorly perfused and appears much smaller and paler than a healthy placenta after delivery. The decidual plate forms these wedge-like projections on the maternal side of the placenta. So when you, as a clinician, come to inspect the placenta after birth, you will be able to see these septations on the maternal side of the placenta as a result. These are known as cotyledons. And this is the overall outcome. Again, use this image as inspiration for your own sketch. And this is how it looks in practice. During the second month, we start to see a bulge in the uterine lining called the decidual capsule, pushing its way into the lumen of the uterine cavity. As the fetus continues to grow, the villi encircling the embryo begin to modify, and those on the lumen side of the embryo begin to smooth, whereas those on the uterine lining bound near the decidual plate remain frilly and retain their ability to exchange with the maternal circulation. The ones that remain frilly are called the chorion frondosum. Placentation is very important, and when it goes wrong, not only is it detrimental to the fetus, but also to the pregnant person. When we perform ultrasound scans, the placental development is considered. We look at the location of the placenta to ensure it is nice and away from the cervix. In the event it is not away from the cervix, we have a condition called placenta previa. When you think about it, having a highly vascularized placenta covering the exit route for the fetus can be quite problematic. Not only do you have gravity acting on a 3.5 kilo baby bouncing on a highly vascularized structure, but you also have the problem of how you get the baby out, as the baby needs to be delivered before the placenta can detach. Placenta previa should be diagnosed via ultrasound scan, as it is one of the things screened for during those scans. If spotted, the baby would need to be delivered by C-section. If you have read This Is Going to Hurt by Adam Kay, the case that drew the final straw for him was how having an undiagnosed placenta previa almost cost his patient their life through maternal hemorrhage. The size of the placenta is also monitored throughout pregnancy, as is the circulation and the vascularization in it. Particularly towards the end of pregnancy, past the expected delivery date, the midwife or the ultrasonographer will use a Doppler to ensure the placenta is still functioning efficiently. For further reading on this subject, have a look at the following article. So let's bring this back to efficiency of the uteroplacental exchange and have a look at what else happens to improve the ability of products to cross the membranes. There are about 100 spiral arteries delivering 150 ml of blood to the intervillous spaces or the lacuna, which is replaced about 3 to 4 times every minute. So the sheer volume of blood moving in and out of those intervillous spaces is incredibly high. As the pressure drops in the spaces, the blood enters and returns to the endometrial veins and continues to circulate around the maternal vascular system, taking metabolites from the fetus. As mentioned, there is no mixing of fetal and maternal blood, and that is because we still have a barrier of syncytial trophoblasts and cytotrophoblasts and some connective tissue and epithelium. However, to modify this even further and decrease the amount of barriers the nutrients have to cross, the syncytial trophoblasts are able to apoptose and slough off into the intervillous spaces and enter the maternal circulatory system. And if you think back to prenatal screening, that cell-free DNA circulating around the maternal circulatory system has been exploited for determining whether or not there are any genetic abnormalities. So this slide shows a healthy placenta delivered shortly after birth. This occurs about 30 minutes after the delivery of the baby, but sometimes this might need to be helped along by the administration of Pitocin, if it's a little slow to emerge. Your job, as mentioned, is to inspect this after delivery and ensure that it looks healthy and intact. But what does a healthy placenta look like? Well, the healthy placenta will be approximately 20 centimetres in diameter and will have a centrally inserted umbilical cord, approximately 50 to 60 centimetres in length. If it is too long, the fetus can tangle themselves up in it, and if it is too short, it can make the fetus difficult to deliver. But let's assume that the baby has been delivered safely and you are now inspecting the placenta. You are looking for the fetal side of the placenta to be covered in amnion. But when you turn this over to the image on the right, you hope to see an intact maternal side. A healthy placenta like this one should show 15 to 20 smooth ridge cotyledons formed from the decidual plate. Each cotyledon should be smooth on the surface and not missing any tissue, as this might indicate that some of it has resided within the uterus. As mentioned at length, the maternal and the fetal blood are not exchanged. But in the event placental membranes are damaged and the mixing of blood is allowed, this could be incredibly problematic, particularly if the mother is of rhesus negative blood, but the father is rhesus positive and the fetus has inherited that. This can lead to hemolytic disease in the newborn. Essentially, the maternal immune system detects the red blood cells positive antigens of the baby and mounts an immune response to that antigen. Consequently, those antibodies can cross the placenta and end up in the fetal circulation and bind to the red blood cells of the fetus, marking them for destruction. The pregnant person is blood typed earlier in pregnancy, and a prophylactic serum is given in the event they come back as rhesus negative, to prevent an immune response being mounted against the fetus if those membranes are damaged. So as the fetus is incapable of breathing or eating, the circulatory system of the fetus is different to that of a grown adult. The fetus has shunts in place to allow for bypassing of the pulmonary circuit and to allow the whole placental circulation to continue. We will look at some of these on the next slide. Oxygenated blood is carried from the placenta to the fetus by the umbilical vein, which will drain into the inferior vena cava through a ductus venosus or the liver. When oxygenated blood enters the IVC, it moves in parallel with the deoxygenated blood from the systemic veins, establishing a bilaminar blood flow as it enters the right atrium. The fetal heart contains 2 upper atria and 2 lower ventricles, just like the adult heart. However, it also contains 2 additional structures, the foramen ovale and the ductus arteriosus, that function as shunts for the oxygenated blood. The function of these shunts is to bypass the lungs and maintain proper circulation to the important fetal tissues. In the fetal stage, the lungs fill with fluid and collapse because the fetus is within the amniotic sac, and the placenta is providing the oxygen that it needs to grow. With the lung collapse, pulmonary vascular resistance remains high during the fetal stage to prevent blood flow into the lungs. As oxygenated blood arrives at the right atrium, the eustachian valve opens and directs the oxygenated blood into the foramen ovale, an opening between the right and the left atrium. As the blood flows through the left atrium, it will move through the mitral valve and into the left ventricle, where it will be pumped through the aorta to the body. The shunting of oxygenated blood from the right to the left atrium, will supply blood high in oxygen and nutrients to the upper extremities, which include the critical organs such as the brain. Some of the blood moves from the aorta through the internal iliac arteries and to the umbilical arteries as it re-enters the placenta, where carbon dioxide and other waste products from the fetus are taken up and enter the maternal circulation. Some of the blood entering the right atrium does not pass directly into the left atrium, but instead enters the right ventricle. This blood consists of oxygenated placental blood and deoxygenated blood returning to the fetal circulation. This blood is pumped into the pulmonary artery. At the pulmonary artery, it is met with high pulmonary vascular resistance as a result of the collapsed lungs and the pulmonary capillaries. In the fetus, there is a special connection between the pulmonary artery and the aorta called the ductus arteriosus. Because the aorta has low pressure than the pulmonary artery, most of the blood flows across the ductus arteriosus away from the lungs. Once the blood goes through the ductus arteriosus, it mixes with the blood from the aorta, and this results in mixed blood oxygen saturation that supplies most of the structures in the lower half of the fetal body. If you wish to approach further reading on this subject that we have looked at today, please have a look at this paper and let it inspire you. And that's it for this lecture. You should now be able to give an account of the functions of the placenta, give an account of the formation of the maternal and fetal components of the placenta, and describe the appearance of the placenta at birth. You should be able to describe the placental circulation with particular reference to the separation of the maternal and fetal circulations. Give an account of the normal fetal circulation, and give examples of problems arising when fetal shunts fail to close.